Thank you so much, everyone, for coming to my talk. Also for inviting me to be here. This is my third time in Colombia, and every time I'm struck by how welcoming everyone is and how much heart you put in crop protection and the research leading to good crop protection research methods. So I will be talking about my research, about the research that the people that I'm collaborating with are doing. It's a lot of collaboration with the Ontario Ministry of Agriculture. It's a lady called Sarah Jendrisic. She has great resources as well that I will point you towards at the end of my talk. And my whole lab and team, the technicians, the scientists, they have generated all the data that I'm going to present. At my position right now, I'm now the director of the Biological Crop Protection Program at Vineland Research and Innovation Center. And I'm in charge of coming up with like all these research ideas and the funding. And I'm so excited to see what the researchers in my lab can do with all that material. So today I'll be talking about integrated pest management for diverse Thrips species in greenhouse ornamentals. And I'll be speaking from the Canadian perspective, but I hope there's a lot of parallels, and I'll try to draw those parallels with the Colombian situation. Just to start with the differences, I guess. So I'm from the Niagara region, where this big red arrow is pointing up north in Canada. It's kind of the most southern part of Canada, so we do have warm summers. Everyone thinks of Canada as being cold, but actually I think today it's warmer in Canada than it is here today. The province of Ontario is the fourth largest floriculture producer after Florida, California, and Michigan. We produce a lot of potted plants like geranium, tropicals, calendulae, some roses like the potted roses, chrysanthemums, and poinsettias. And those are the plants that I work with most. We also produce bedding plants, both vegetable and ornamentals. Those are kind of like the geraniums and all the plants that you plant into your garden in spring. And then we produce a tiny bit of cut flowers. Most of our cut flowers you are growing for us, so thank you. But we do produce tulips, some gerberas, some chrysanthemums, and things like snapdragons. And then a little bit of the industry is producing cuttings, propagative material. So in Canada, we're very lucky to have a very high adoption of biological control. In both vegetables and ornamentals, it's the main strategy that growers use to control pests, and about 50% could use biocontrol to control diseases. It really began in the 1990s, where we had problems with that we had very few registered pesticides. So it was very difficult to rotate modes of action. And insecticide resistance developed in all of our common pests, like the thrips, aphids, spider mites, and some of the others. And we have our most challenging pest in common. Western flower thrips is also our most challenging pest. And then in second place come aphids, and I think that's the same again. We have very similar aphid species that you are battling, and spider mites and mealybugs, depending on the crop, come in third place. So I've been working on Western flower thrips integrated pest management or biological control for a very long time. For most of my career, it's kept me employed for about 20 years now. So I am very thankful to Thrips in a way that they have allowed me to have a job. My job, though, was to kill the thrips that keep me employed. So with biological control, and this is kind of a representation of all the research that's gone into how to develop a good biological control program for Western flower thrips. So a few years ago, I was very proud to say, like, look, we have solved Western flower thrips. We use predatory mites, basically from start to finish, We use cutting dips. We do other biological control agents like here in the soil, nematodes, biopesticides, so that at the end of the crop cycle, we maybe have to use a pesticide, or maybe we're lucky and we don't have to use a pesticide. Of course, it's too good to be true. Just when we thought that we had stuff figured out, we did a survey of growers, and about half of them said, like, well, I'm not that happy with my control program. And also, more than half of them experienced more severe thrips outbreaks. So we were not, obviously, there was a problem. And we were asking ourselves, what changed? And some very bright people came up with the idea, well, maybe we're dealing not only with Western flower thrips, but with different thrip species. But no one had ever looked since the 80s, since mid-80s, what thrip species we actually had in our Canadian greenhouses. And me included, like when I did my research, you know, you'll see a thrips and you're like, oh, it's Western flower thrips. So no one really actually looked at what species were there. So my colleagues from the Ontario Ministry of Agriculture did a survey, and this reflects the Ontario situation, but I think there are a lot of parallels with you. So most of the thrips, 65%, were still Western flower thrips. However, we saw that about 30 to 35% were onion thrips, Thrips tabaci. And then there were a few others like echinothrips and some other species. And this was consistent. Of course, the proportions varied in different crops, but we also saw this in vegetables. And the other thing that was happening is that there were more and more invasive thrip species coming in with plant material, like increased global trade. In Canada, a lot of tropical foliage plants became really popular when everyone was isolated due to COVID. Everyone wanted to be a plant person. We had another wave of deregistration of pesticides. Climate change maybe also makes it easier for some thrip species to establish at more northern latitudes. And also, if you start looking for things, you will find things. So just knowing that they may have been there already, but no one ever looked for them. And here's a special case, and I'll talk about especially Thrips parvispinus a little bit more, but these look basically identical. So sometimes people said it's Thrips parvispinus, but it was actually thripsitosis. So what changed? I think I touched on a few things already, but making the switch from a main pesticide program to a biological control program over the course of 20 years, I think we basically lost our selection on Western flower thrips. Western flower thrips are really good at being resistant, while the other thrips are often still susceptible to pesticides. So if you spray a lot, you will have a lot of Western flower thrips. If you stop spraying, you may get a more diverse thrips population. And what we saw and why we had more severe outbreaks of other thrips is that our biocontrol program that I showed you, that beautiful infographic, was specialized to Western flower thrips and may not have worked as well against the other thrips. So knowing the thrip species that you have in your crop is very important for the choice of your control program. First of all, if you want to use pesticides and you have mainly Western flower thrips, it will be very difficult to find some products that still have high efficacy. However, like I said, the other thrip species may still be susceptible to pesticides. Also, if you want to do a biological control program, like I said, the program that you do for Western flower thrips is mainly based on predatory mites and some biopesticides, may not work as well for the other thrip species. So we are now doing a lot of research and again, thrips are keeping me employed, we're looking for programs that can manage a complex of thrip species. Now, of course, I know Canada is very different from Colombia, but we have a lot of thrip species in common. And here I try to kind of put the main thrip species. So we have western flower thrips, Thrips tabaci or onion thrips, echinothrips, and chrysanthemum thrips, Thrips nigropilosus. And then we have some new invasive ones, of which Thrips parvispinus is causing the most damage at the moment. You also have western flower thrips and onion thrips. And then I understand that you also have Frankliniella panamensis, which is also occupying a high percent of your crops. And other invasive species also include thripsitosis, for example, in hydrangeas. That can be a big problem. So there are a lot of parallels. The stuff that I'm going to present, of course, applies to the Ontario situation in Canada, but I hope that it will give you some inspiration for what you can look at here in Colombia. So we'll start with monitoring. And like I said, know what thrip species you have in your greenhouse. Learn to identify the most common thrip species in your crop. And we have set up some workshops in Ontario with our most common thrip species for growers, and we encourage them to buy a microscope. Of course, the lab microscopes that we use at the university and at the research station are very expensive. But nowadays, you can buy a microscope that's very similar for about $400 American on internet. So a microscope should not break the bank. To identify thrips, you have to look at things like these little hairs here and the little ocelli and that kind of stuff. And you need a microscope that with a 40x magnification for that. So any of these will do this. And this is a screenshot of a video that we made. And I'll give you the link at the end as well, how to choose a microscope that will do the job. So there's several ways you can monitor for thrips. And here I put the most common ones. You can use sticky cards. You can use plant taps where you have a white surface and you just take the plant and you tap it above the surface. You give it a good firm taps. And then you can do something that we call plant washing, where you collect parts of the plant and you wash it in either soapy water or ethanol. And again, this is a video, and I will give you the link at the end. Another tip is if you are dipping cuttings, and I've done a lot of research on dipping cuttings, and we'll touch a little bit on that as well. But if you're dipping cuttings, you can actually filter the water that you have dipped all your cuttings in and see if anything washed off as a first indication of what may be coming in on your cuttings. And then, of course, different thrip species and different pest species will have different symptoms on the plants according to their damage and where the damage is located. So going a little bit more in depth on the different monitoring methods, on this slide, I'm comparing sticky cards versus the plant taps. And it will give you very different information. I wouldn't say that one is better than the other. You just have to know what it represents. So we, at different sites for different thrip species, we did either plant taps in red or sticky cards in yellow. So I'll first talk about western flower thrips. You see that with the plant taps, we got way more western flower thrips than with the sticky cards. For this species, which is eastern flower thrips, and I don't think you have that, we actually saw more individuals on the sticky cards than from the plant taps. And for onion thrips, it depended on the greenhouse. We have 3 different sites, and only in one site we found more in the plant taps than on the sticky cards. So what this means is that depending on the monitoring method, you will see different things for different species. Sticky cards will tell you what kind of thrips are flying around. Of course, it's obvious, right? The thrips are attracted to the color and they get stuck. So only flying individuals will be caught. The plant tabs will actually tell you what species are on your plants. And like I illustrate here, it can be very different. And then comparing the three methods, including plant washes. And I think the biggest difference that you can see already is that plant washes, you will find way more thrips than with any of the other methods. Here we were looking at Thrips parvispinus, larvae in red, females and males in these two colors here, and other Thrips species. And you can see really that the cards and the tabs kind of gave equal numbers, while the washes gave you a lot more numbers. And the other thing that is very typical of plant washes is that you finally will be able to get to see the larvae. The larvae of thrips will not get caught on sticky cards. Obviously, they're not flying. And plant tabs, somehow they hold on better to the flower or the plant tissue, so you don't get as many larvae off as with washes. So with washes, those are the only methods that can reliably show you thrips larvae. Of course, the whole debate, sticky card color, the debate has not been solved, by the way, but different thrip species probably have different color preferences. And we'll talk about this later when I talk about mass trapping as well. But yeah, we did a quick trial with parvispinus, females, males, and other thrips, and looked at yellow, white, and blue sticky cards. And in this case, we did not find a difference between the yellow and the blue sticky cards, while the white cards performed a little bit less. This is actually interesting. There's a company in the US called Catchmaster, and they will make sticky cards any color that you want. That's kind of interesting. So here, a grower had purple flowers that they saw was very attractive to thrips, and they made sticky cards that color. So when someone tells me about their greenhouse and that they have thrips, my first question actually is, do you know where the thrips come from? Because knowing where they come from will tell you what biosecurity methods you will have to put in place. There's several options. They can come from within your greenhouse. You can have a population in older plants, and this arrow should actually be turned the other way. So the thrips can go from your older plants to your new plants, which you, of course, don't want to. The other option is that they are flying in from outside. We already talked a little bit about that. Or maybe they come in on your plant material, which is another option. And we'll talk about all of these. Like I said, the source can be your own farm, so try to start clean as possible. Disinfect your compartments in between cycles. Try not to mix old and young plants, where the older plants are more likely to be infested than the younger plants. If you clean up and you grow in soil, try to clean up the soil as well, maybe with some solarization, steaming, or even using a microbial a microbial application in the soil. And also have your workers be aware of the order of entry. In my lab, if you have been in the thrips colony, you are not allowed to go into the clean plant room. Similar to that, of course, if workers have been in an infected greenhouse, then they should not go into a clean greenhouse. Let's talk a little bit about the fly-ins from outside. And I already gave away a little bit of the punchline here, but there are differences between thrip species, where they come from. We monitored sticky cards inside and outside greenhouses. And again, this is for Canada, but you can do the same here in Colombia. And there was definitely a correlation between the inside, which is the red line, and the outside, which is the blue line, for Thrips tabaci and the eastern flower thrips. which told us that they are most likely coming in to the greenhouse from outside. This was not the case for Western flower thrips. There was no correlation between the inside and the outside cards, and there were way more Western flower thrips inside than outside. So that tells us that the Western flower thrips probably, they may have come in from outside, because yes, we do have Western flower thrips also in Canada outside. But there may have been other sources like plant material that brought in the Western flower thrips. So what can you do? We, again, have a big debate about screening. Some people love screening. Some people say, like, I will never screen my greenhouse. But screening is the easiest way to put a barrier between the outside and the inside. And if the mesh is small enough, it will exclude even things like thrips. There is some research that different colors or the different optical properties of screens may exclude thrips, even if the mesh size would theoretically let them in, such as red screens, but I don't have time to go into that for here. But the downside, of course, is if you screen your greenhouse, you will also exclude free natural enemies coming in from outside. However, I think there's a lot of technology out there now. If people say, like, I can't screen my greenhouse, there's some people here at the Proflora show that will have some technology where you can talk to them about how to maintain the airflow in your greenhouse while at the same time excluding thrips. Again, coming back to sticky cards, you can also use sticky cards not only for monitoring, but for mask trapping. Any thrips that's stuck to a sticky card or a sticky ribbon is one less thrips. Do you use blue? Do you use yellow? There's very conflicting studies. So basically, my message to you is try them all out in your greenhouse, because the greenhouse covering and other properties of the greenhouse will have an influence on what works better. Overall, yellow is more multifunctional. It will not only catch thrips, but also other pests. So a lot of people prefer yellow for that. But yeah, just test them in your own facility if you want to know what works at your site. Trap plants may also be an option to intercept dispersing thrips. Trap plants will not, mask trapping and trap plants both will not remove the thrips when they're already on your crop. Again, they remove the dispersing or the flying thrips that are moving through the greenhouse. You can place trap plants close to vents or doorways or between crops. Here we use flowering mums as a trap plant in non-flowering mums. This is alyssum in a strawberry field, and it basically creates a buffer that intercepts the thrips before they go into the crop where you don't want them. If you're really courageous, but this is a great idea, I think, but it has some potential for disaster as well. But I would like you to think about creating actually a beneficial habitat outside your greenhouse that will intercept the thrips, kind of act as a trap, but at the same time attract beneficial species and become kind of a breeding ground and your standing army for protecting your greenhouse. So in the Netherlands, this research from Gerben Messelink and his colleagues, they wrote a wonderful paper about it if you want to know more. I think this could be very interesting because here in Colombia, you must have also a lot of naturally occurring beneficial insects. And why not have them do the job for you? The other source of thrips can be important plant material. And I'll give you two examples here. This is the example of Western flower thrips. And we just try to figure out how many Western flower thrips come in on chrysanthemum cuttings in each shipment. We looked at a lot of different varieties. So each of these bars is a different variety. And we looked at them the day they came in, it's the orange, and 6 days later to allow the eggs of thrips to hatch so that we can actually see them. Because when they're in the leaf tissue, we cannot see the thrips eggs. So the red bars basically represent the thrips that came in as eggs. And this is quite scary, actually. I think most of them were eggs and larvae. There were big differences between varieties, but also over the course of time, it varied by week and supplier. But basically, if you are bringing in 10,000 cuttings per week, which is not a lot, I know greenhouses that do way more, but if you do 10,000 thrips cuttings, you may carry in 400 thrips in your greenhouse. And those, of course, will reproduce and get more very fast. With Thrips parvispinus, our main nemesis now, it's very similar results. Thrips parvispinus comes in on tropical plant material like Dipladenia or Mandevilla. And big differences between varieties. Sometimes the white ones had a lot of thrips, while the red ones did not have any, and vice versa. So again, this thrips comes in on the cuttings. And again, 10,000 cuttings would amount to about 100 thrips. So luckily, we've done a lot of research on cutting dips. And I've talked about this before at previous Profloras, so I'll just skim over it a little bit faster. But cutting dips are basically immersing your cuttings in a solution of a reduced risk product like oil or soap or a biopesticide. It will kill pesticide-resistant thrips, which is very important because at the propagator, they probably have a very stringent pest control in place, which may have contributed to the development of resistance. These products will leave minimal residues so that you can use biological control if you want to after on these cuttings. And then you dip them, you stick the cuttings and continue as normal. What this does is you are reducing the starting population of thrips so that you give your biological control or your other IPM program a much higher chance of success. This graph illustrates if you do nothing, your thrips over 4 weeks, they will go through the roof. If you use biological control only, so no dips, the thrips may go up because they have a hard time actually getting the upper hand on the thrips. While if you dip and then do nothing, the dips initially will lower the number of thrips, but there will be a few left, which if you do nothing, again, it will go up. So if you combine dips and then do biological control, that's the best recipe. So dips basically give you a head start, but you have to do stuff afterwards. This is a huge table. I tried to summarize all the different experiments that we've done, and there's a lot of empty cells there that we just don't have data for. But in general, you can see all the materials that we have tested, and there's other labs and other groups that have tested materials as well. So this is not all the information that's out there. But we are looking at the different materials for Western flower thrips, Thrips parvispinus, Bemisia tabaci whitefly, and spider mites. So you can see there are products that will work for multiple pests. For example, if you have all these pests, a product like soft oil, which is a mineral oil, will be a very good choice. If you have only thrips, you may want to go with something that has maybe a little bit less risk of phytotoxicity, such as a wettable powder of a biopesticide. These symbols kind of indicate that a little bit of caution is necessary because we saw some phytotoxicity with some plant species. So that's what that means. On other plant species, it worked really well without any phytotoxicity. Going into a few alternatives to dipping, some people were critical of dipping because they're afraid that if you dip, you will transmit diseases like soft rot or other pathogens in the dip water. So here's a few alternatives that actually work really well as well. This is a company from Canada that combines UVC light, hydrogen peroxide, and ozone in the same treatment. It's kind of like a tunnel or a little robot that can drive over your plants. And it produces radicals, which basically kill the insects or even some pathogens. So preliminary trials, we put some plants with onion thrips through the machine and saw that we got 50% reduction of thrips on those plants. So that was really promising. We did it with rooted and unrooted cuttings, and the research is not done yet, but there is some promise. Basically, only the high dose of this method worked against eggs, while the medium and a high dose worked well against thrips larvae. The other thing that we saw that sometimes there was some phytotoxicity, so we still have to figure out why that is and if there's a dose that is safer and more effective. So we also want to do future work with that little robot where we can maybe drive over the plants and apply this to whole plants. Interestingly enough, Thrips parvispinus is a tropical thrips. So I hope you never get Thrips parvispinus, really. But if you get it, it is susceptible to cold. And no other thrips that we have does this. So we can actually kill Thrips parvispinus eggs, larvae, and adults by exposing it just to cold for several days. So we took the cuttings, and these are mandevilla cuttings, and we exposed them to cold for 1, 2, and 3 days. And you can see towards the 3 days at about 4 or 3 degrees Celsius, we did kill a lot of thrips. If the cuttings can be stored at these temperatures, this is a very simple method to get rid of them, but it will only work for parvispinus. Western flower thrips happily come to life again, even after you put them in the freezer, which is very frustrating. The opposite of cold is hot steam treatments. This is a picture of a company from the Netherlands. It's called plant sauna, and that's really what it is. They put plants in a sauna, and with steam, they first raise the temperature to 37, then they give the plants a little bit of a break, and then they heat them to 40 degrees Celsius. And this will also help disinfect against pests. We had a lot of problems with oleander aphids on our mandevilla mother stock plants, and we killed them all. We put them all in the sauna, and now we have nice, clean mother plants. But of course, you can only do this with plant material that you are able to transport. It won't really work in a greenhouse. So the main message about cutting treatments is you can prevent thrips infestations if you know that they come in on plant material by treating the cuttings and plugs. Which treatment you use depends really on the crop species and the stage. And I can't stress it enough, please always test it first on a small batch before you go all in, because I know some growers who actually came back to me and said, you said soft soil was safe. And I'm like, I also say, always test on a small batch first. We'll make it to the end, hopefully. So I'd like to talk a little bit about host plant resistance. Not all plants that may look alike are equally susceptible to pests, to thrips, for example. Here we have several yellow garden mums, and as you can see, in the vegetative stage and in their budding stage, there were big differences in the number of thrips that we found on them. We also did some biochemistry on these plants, and we saw that there were differences in biochemistry. And we think we have found some biochemical markers that correlate with Thrips resistance. And I've been talking to a few of the breeders here at Proflora about this, that we should actually continue this research to see what those markers are. And we don't only see it in chrysanthemums, we also see it in mandevilla. Here you see a not susceptible variety for Thrips parvispinus, and these plants are absolutely devastated by parvispinus. Same with anthurium, beautiful anthurium varieties, no problem with Thrips parvispinus, and these are very much eaten. And even in greenhouse pepper, sweet pepper, there are differences. So what we're proposing is that we collect a lot of information on Thrips resistance that's not available from breeders because they don't know exactly, they've never really looked at it. In your greenhouse, you can keep records, which varieties get Thrips the most. But what I really would like to do is screen a lot of different varieties or different genetics through the metabolomic analysis, and then you get kind of like this kind of fingerprint of what biochemicals are there, which we will be able to tell based on those markers which plants will be more or less susceptible to Thrips. And then lastly, like I said, Western flower thrips, we had a whole biocontrol program and which did not work for onion thrips or for Thrips parvispinus. So again, biological control programs, adapting it to the Thrips species or the Thrips species complex that you have is very important. And these Thrips species even differ in their daily activity. So over the course of a day, Western flower thrips is kind of active in the morning and then active again in the afternoon. Onion thrips are active in the morning, and I don't know what happened to this graph, but Thrips parvispinus are active in the afternoon. So once you know what Thrips species you have, and if you are relying on products that rely on contact, like biopesticides, you should adapt the time that you're spraying these products to the time of the activity of the thrips. Also, Thrips species have differences in how they react to biocontrol agents. The Western flower thrips are mostly controlled by predatory mites in a biocontrol program, but thrips may fight back. Like they make these little droplets that they basically just fling at their predators as a defense. They may eat the predatory mite eggs. And as you can see, the relative difference, if this thrip starts to struggle, this predatory mite will have a very hard time of controlling it. So depending on what species you have, you may want to include more bigger bios or even microbials that do not have these problems. The different thrip species also are located in different places on the plant, and that's very well illustrated by this picture here. This is two plants that are exposed to the same number of thrips, but these were onion thrips and these were Western flower thrips. You see that the Western flower thrips really like the flowers, while the onion thrips mostly were on the foliage. And other foliar thrips like chrysanthemum thrips and echino thrips also have a similar pattern. So again, where you apply your biocontrol or your sprays or whatever you use, you may want to adjust that depending on the species that you have. This is 3 years of research in one graph, but I just want to illustrate that for onion thrips, we finally compared the commercial standard for Western flower thrips based on predatory mites and some microbials to a bugs only, a microbials only, and a mixed model. And you can see there some of our new programs definitely work better for onion thrips than the standard Western flower thrips program. For parvispinus, we don't have the answer yet, but we think one of the answers is in the bigger biocontrol agents like Aureus. And we have this big predatory mite called Anistis baccharum I'm not sure if you have it, but it's a relatively new biocontrol agent. This is not Persimilis. It is red, but it's much bigger than Persimilis. And again, here you can see that both these biocontrol agents could suppress Thrips parvispinus quite well. So we're pretty happy about that. Of course, what crop you're using has an effect on how well your biocontrol agents go. So a biocontrol program for sweet pepper will maybe not work in chrysanthemum and vice versa, right? So make sure that you know what works in a specific crop. And then finally, here are my recommendations. I hope that I gave you an impression of what to look for based on our experiences in Ontario, Canada, that you have to look for the different thrips species that you have. Learn to identify them. Determine how the different species actually get into your crop so that you can adjust your biosecurity methods and also adapt your control methods to the species complex that you have. You'll probably have different species at the same time in your greenhouse, so that makes it more complex as well. So consider pesticide resistance, their behavior and biology, and also if you can grow resistant crops or plants that can tolerate more thrips damage, that would be fantastic. So just to point you to some of the resources that I alluded to in my presentation, if you go to my LinkedIn profile, so I see everyone take pictures. If you go to my LinkedIn profile, I made a post with all these links. So if the picture doesn't work for you, then please look me up on LinkedIn and you'll have all these links here. Also, of course, Laura already told you about the Thrips and Botrytis resources. So here's another QR code to that. And I would like to thank you very much